Thank you very much for the kind introduction, for giving me the opportunity to talk a little bit about natural products research in drug discovery. So just a brief background. So we are just in the process of building up a new research group in Vienna, which is focused on cheminformatics, in particular machine learning. The work that I will show you today is mainly coming, mainly the results of Ya Chen. She's basically specialized in natural products research. Our research group develops new approaches in early stage drug discovery. So you see here a little bit of an overview of the types of methods and questions that we are working with. And the red ones indicate where we have some active method development going on, and the blue ones are the ones where we employ computation methods to in the context of academic, mainly academic drug discovery. So in collaboration with national and international collaborators. So what we do a lot is actually virtual screening and everything around hit identification, and then also hit to lead and lead optimization. So once we have interesting compounds, we go into more sophisticated methods to also understand, for example, ADME properties and toxicity properties. We also try to identify the likely targets of these compounds beyond those core targets of interest. And we use structure-based methods and structural information whenever possible, but often we also work without it when there's no structural information available or derivable. And as it happens, we are in a lucky situation to be able to collaborate with a lot of scientists that are engaged in natural products discovery, natural products drug discovery. And so we are regularly also screening together with them natural products databases and optimize natural products or derive natural product mimetics. And today I would like to briefly show you how these cheminformatics methods can be employed in this particular context. So it may be a bit surprising that even for modern small molecule drugs, more than 50% are related to natural products. So we see this here in this nice diagram, where we basically see that these green areas and the blue areas, they represent compounds, so proportion of drugs that are related to natural products. So they are not necessarily natural products, genuine natural products. These are only 5% actually, but natural product derived compounds are plenty among the drugs. So we have some that are derived directly from natural products, so they are basically derivatives thereof. But many of them are also inspired in terms of the pharmacophore or the structural templates. And only 1/3 of drugs are entirely of synthetic origin. But to be fair, though, when we look more closely into these data sets that are being published in these famous works, actually, if we look more closely to that, we see that these natural product derived drugs, they are not equally distributed across the chemical or the drug space. So we have mainly compounds there that have higher, larger molecular weight, typically also often higher than 500 Daltons. And we have an overrepresentation of some prominent scaffolds, such as steroids or macrolides, in particular rapamycin analogs. So why are natural products so interesting for drug discovery? Now the answer is simple and can be intuitive. For example, we think about plants. Plants simply can't run away from, for example, other organisms that want to eat them, or if you think about fungal infection or caterpillars or sheep and cattle that want to eat them. So these plants, in order to protect, for example, their leaves from being eaten, they need to have some toxic flavor or taste hot or bitter, or they need to actually be toxic. And so what we know, of course, is that these natural products, they have been undergoing optimization throughout the whole course of evolution to basically exhibit some biological activities. And as it happens, these biological activities or related biological activities are often observed in other organisms. And the reason for that is actually that protein structures within different organisms may be fairly conserved. So it is not surprising that some of these natural products, or quite many of them, are actually based on privileged scaffolds. What are privileged scaffolds? Privileged scaffolds are molecular or ligand cores that enable this molecule to bind in a specific manner to several different proteins. So these are basically molecular frameworks that are particularly interesting for medicinal chemistry because they have a high chance of being active on some targets, and they could be valuable starting points for an optimization. Then this optimization, of course, needs to also involve the optimization of selectivity towards the intended targets. So a big challenge in natural products is actually that they have higher hit rates in biological assays than traditional synthetic libraries. So this is good on one hand, because you actually are more likely to find something, but it's also a big challenge because some of these hits may not necessarily relate to genuine biological activity. They may be more related to assay interference, and the underlying mechanisms can be very diverse. So, but I already mentioned the protein structures between different organisms, they are often similar. But, and for this reason, plant constituents, for example, may trigger biological effects in humans and other species. But the interesting thing is that these organisms can differ substantially in terms of, if you think about pharmacokinetics. And this can help us to actually obtain or design compounds, natural product derivatives, that allow us to obtain target organism specificity. Now, if we want to work on with contributors on natural products, then we are facing several challenges. First of all, as a big plus, of course, is that natural products have an enormous variety of physical, chemical, and structural properties. So we see here a comparison. So this is a principal component analysis, which is based on very simple physical chemical properties like log P, molecular weight, hydrogen bond donors, etc. And what you see here at the first instance is, I see a big cloud, a big clean cloud distributed over the whole range. So these are the natural products, and somehow a subspace thereof are synthetic molecules. And these synthetic molecules in this particular case represent the zinc database. So you see at first sight, we have molecules in natural product space that are much more diverse than we generally have in the synthetic molecule space. So with that comes the fact that natural products are often of high molecular complexity. So we have higher molecular weight, often even above the 500 Dalton, that are generally considered the border of not drug likeness, but at least oral bioavailability. We have 3D shape complexity, a very so that means basically we have molecules that are much more complex in the 3 dimensions, which has a big advantage in addressing certain types of binding sites actually. And this comes because we have a higher degree of sp3 hybridized carbon atoms. In particular, something like this here, what you see here is there's like bridged carbon atoms, and they make these molecules really to have complex 3-dimensional shapes. And what we also have, of course, is more complex ring systems, which I will show you in a moment. So when we want to engage in natural products research, then we are, so one of the bottlenecks is actually the limited availability or the costs of materials for testing. Just think about very simple things like, first of all, these interesting compounds may only be available in very low quantities in organisms, such as plants, and that's not enough. It is also very difficult, not just logistically, but also legally, to import or export certain types of natural products. So it's often a very long road to get these natural products actually in the vial, to be able to test them experimentally. And these natural products, they are actually known to be prone to cause false positive hits in biological assays. Just to give you one interesting example, this is quercetin, which all of you may well know. And by checking PubChem Bio database, so a database of biological activities, we find that currently for these natural products, activity data is reported for 427 different proteins. And out of these 427 proteins, 268 of them are annotated with an active label for this particular molecule. So this means basically this molecule, if you take this literally, could be a blockbuster drug and just curate like any disease, which of course is not the case, because many of these activities are actually, or these measured activities are in fact not activities that you would also observe in vivo, but they are rather related to assay artifacts. So when we want to employ computational methods in natural products drug discovery, then we are challenged by the molecular complexity, as I showed you on the previous slide, also on the flexibility of the compounds. Then very good question of course, already by the chair of the session was regarding the stereochemistry. Here we have a really, really big challenge with stereochemistry, for the fact that stereochemistry for natural products is often incorrect or unknown. So we need to be very careful with that. And as many of the methods are actually ignorant of or do not consider stereochemistry. So there's a big challenge involved there. And lastly, computation methods have been devised from and developed for synthetic drug-like molecules rather than natural products. And in order to use them also in the natural product space, some modifications and adaptations may be required. So not too long ago, we actually tried to summarize the state of the art and the scope and limitations of computational methods in natural products-based drug discovery. And in this review, and also today in my talk, I'm trying to cover the most important aspects of where we can actually employ cheminformatics methods in this particular area of interest. So cheminformatics methods are currently, or today, broadly applied in natural products-based drug discovery simply for one reason, one important reason being the fact that we have these precious materials, and we need to make really sure that we select these materials most carefully and be able to focus our limited resources on the most promising natural products. So cheminformatics approaches are today involved everywhere from the retrieval, the organization, the curation, and the management of chemical information. And as we saw already in previous talks very nicely, visualization and analysis. So there's lots of very powerful tools out there. Then a very important point is natural product dereplication, because we just don't want to, investigate the same natural products over and over again. We want to make sure immediately to basically identify compounds that are already known, and we want to basically immediately be able to recognize novel molecules. Then we want to, of course, be able to predict the molecular properties of natural products, identify bioactive compounds, and on a larger scale, not only predict whether a compound is active or inactive on a particular target, but what we actually want is we want to get some feeling about the bioactivity spectra of a molecule. In other words, we want to understand in what target space our molecules are active in. And for this, for example, understand the selectivity of the compounds or the potential of side effects. And of course, we want to also predict the atomy properties and the toxicity properties. This comes along with the fact that often it's actually the metabolites that are toxic, and it's very, very challenging to predict the metabolites correctly of natural products, and then on top of that, assess the toxicity of these metabolites or the natural products as a whole. So there's a big challenge involved in there. Then big topic is, of course, the design of nature-inspired compounds. So compounds that are synthetically more easily accessible. They are less complex, but they, for example, are based on a pharmacophore that we derive from bioactive natural products. A very nice application that I will also show is the quantification of the natural product likeness of compounds, which, for example, allows us to design molecular libraries for virtual screening. And last but not least, a big topic is assay interference prediction. What we actually want is computational methods that can trigger an alert to basically alert scientists about compounds that are likely to interfere with some biological assay technologies. So I will slowly now go through some of these topics. So the first one being virtual screening. So computational methods, in particular virtual screening methods, have a strong track record of success in natural products-based drug discovery, and there is a very broad range of computational methods that can be applied today for virtual screening for natural products. So we have powerful 2D and 3D similarity-based methods in place. We have reduced graph representation in place, where actually Matthias Hari did pioneering work. Then pharmacophore models, molecular shape-based representations, ligand docking methods, machine learning models, and so on. The challenge, as I already mentioned briefly before, is again here the structural complexity of the natural products, the limited quality, the coverage and size of the bioactivity data sets, which we could use, for example, to train machine learning models. These are generally very data-hungry models or approaches. And just to give you an idea, so we have actually the number of natural products that are for which we know, for example, the protein-bound ligand conformation is very small. We only have such data on about 2,000 unique natural products. So overall, we are always actually limited by data. But that said, nowadays there are actually large data sets out there, even in the public domain, that can be downloaded in bulk and used for virtual screening. I just wanted to give you a brief overview of that. And of course, you should not read through all these tables. This is not expected, but you can look up these tables also in these publications from us. What came out just 6 days ago is a publication on the third generation of the supernatural database. That's a really interesting one. It's actually the largest set of natural products, 450,000 natural products, and it has now, as I spotted, even the option for bulk data downloads. That sounds really promising. I didn't have a chance yet to really investigate it in detail, but that looks very promising. Then another well-known resource is the coconut database. But for the coconut database, it's known that it's quite noisy. So there's a lot of synthetic compounds in there too. So coconut does need further curation before use if you really want to make sure that you're only working with natural products, which is not necessarily always needed. And I strongly believe that any of the other databases also has some shortcomings, but for coconut, we know it because we analyzed it in very much detail. Then dictionary of natural products. This is basically a reference data set that has long been the reference standard for natural products, for an encyclopedic natural products database. Problem is that the license fees are substantial. The UNPD was interesting for a long time, but it's no longer available, and so on. And there are some smaller size databases like 50,000 compounds and so on. And they look at, or they are focused on particular kingdoms. So like plants, or a lot about microorganisms, bacteria, fungi, and so on. And interestingly, we have also quite a lot of databases and comprehensive databases out there about traditional medicines. And these include Chinese medicines, Indian medical herbs, and so on. So there's quite some substantial collections out there. Further to that, there are more focused libraries that are, for example, focused on specific habitats like marine life, or specific geographical regions like Africa, or also, of course, the Americas, South America, Brazilian plants and organisms, for example, and also developed at the University of Mexico City, the BioFakim, which is a nice collection of natural products from local plants, fungi, and so on. And all of these, or most of these databases, are actually available for bulk download. So that's really nice because it makes them accessible to virtual screening. Then other databases are also of interest, and they are maybe more focused. So for example, on specific organisms, or specific, for example, activities like natural products that are related to cancer or inflammation, or they are related to specific natural product classes. So there's a lot out there nowadays that we can actually use for screening. And we have in our group very recently employed more and more machine learning based approaches for virtual screening for natural products. And so far we had really very interesting, very, very promising results with that. So this is just very briefly to show you a slide about the coverage of the data sets that are in the public domain versus those that are basically in the commercial domain, where you need to buy the data sets. And the advantage of the commercial data sets, or the edge, or the additional information that is included in these commercial data sets compared to the public ones, is shrinking substantially in recent years. So you see that there's actually a huge overlap between this well-known dictionary of natural products, which is the commercial data set, and the freely available data sets. Nowadays, this part has increased substantially. So we can get very far nowadays with public data sets. Now, this here is a very interesting number. This is the number of natural products that are readily purchasable. So this means what we did here is we overlaid the off-the-shelf compounds, so basically those that are immediately, or that should be immediately available for purchase, and compared them to the known natural products. And out of them, you see that the number of available natural products that you could order today and hope to get within 2 to 3 weeks to test in your lab is about 25,000. So this corresponds to approximately 10%, which is actually not much. But the good thing is, this is only when you consider the exact matches. So these are exact matches of molecular structures. But if you say, I'm fine with compounds that are structurally close related to natural products, so basically natural product derivatives, then you can get about 25% of the known natural products covered. So 1 out of 4 compounds you can readily purchase, something like this. So in the range of that, which is, I think it's not too bad of a number. And interesting thing that we found is actually that many of these natural products are available from providers that are not necessarily well known. And interestingly, several of them don't even mention anything on their website about the fact that they are providing natural products. This was a very interesting observation for us. And so what we decided to do is we developed machine learning models. We developed machine learning models that allowed us to actually identify natural products in such commercially available data sets just by structure, so that we don't basically need to rely on the labeling by the compound providers. Because we saw also the opposite effect, right? We saw also the opposite way that, for example, providers claim that their compounds are natural products, but this is not necessarily true for all of these compounds that are included in these collections. So what we developed here is a very simple machine learning approach, basically. An approach that is based on molecular fingerprints. So these are random forest models that are trained on synthetic compounds and on natural products. And we develop basically classification models that can identify these natural products with high accuracy. And the way that we reach this high accuracy is that we put enormous effort into sanitizing, in particular, the natural products databases. Because in these natural products databases, as I mentioned already, there happen to be quite a substantial number of synthetic compounds hidden. And we need to first make sure that we reduce this proportion of synthetic compounds in the natural products dataset to the furthest possible extent. And the situation is actually also similar for the synthetic compounds. And also among them, there are lots of natural products. So lots of tricks and know-how went into reducing the noise in these data sets. And once we had these very nice data sets available, we got also very nice prediction results. These are the classification results here on a test site. So this is holdout data. This had not been used during any stage of the generation of the models or the development of models. And you see that here, and so it's the Matthews correlation coefficient, which basically is a measure for the goodness of a binary classification in the area under the curve, is a measure of how well the natural products can be identified correctly and ranked highly in a rank ordered hit list. And you see that, so they basically, both measures go to 1.0, and you see that both measures are very close to this ideal case, which means that these models apparently do a very good job in identifying the natural products. What it also shows, though, is that this task is not overly difficult. So these models work particularly well, and you can run them over any data set to filter out natural products, to identify natural products with high accuracy. Now, why is this interesting? It is interesting if you work with data sets that you want to use as natural product data sets, for example. I just want to give you one example here. We just need to be careful here. So this is a logarithmic scale in all cases. So what we see here first is the classification on a test set. And on the test set, we see that this classifier really works well, and it can distinguish the natural products here in green very well from the synthetic molecules. And there's only very few, only 1% of the compounds are assigned a probability value close to the decision threshold. So which means that here we have some uncertainty about into which class these molecules may fall. And it's also very interesting to see this in the case of the Campbell database. The Campbell database provides a data set. This is called the Journal of Natural Product subset. And this subset is actually used regularly to train natural product likeness scores. But when we investigated this data set with our natural product with NP Scout, with our machine learning models, then we found that actually there are some molecules in there that are predicted as not drug-like at all. And by looking at them, we realized that this data set actually contains a substantial number of reactants that are of purely synthetic origin. So basically, this is not, as the name might suggest, a data set purely based on natural products, but it's based on any molecules that are reported in this journal, which happen to also, of course, include synthetic compounds that are used as reagents, for example, in biological assays. And models like this can help you very easily to curate your data. And on the top of that, you can use these similarity maps, which allow you to basically visualize which atoms in a molecule were decisive for the classification of a compound. So here you see, for example, in green, you see parts in a molecule that are obviously related to natural products and other parts that are related to some more synthetic chemistry. And yeah, with this, you get a very nice idea of why a model has predicted a certain compound or assigned a certain class label to a compound of interest. And lastly, I just want to show you very briefly an interesting study about target prediction. So what we were interested in is actually to quantify the success rates of the following common problem. So in online databases such as Campbell database and PubChem Bias database, you have millions of compounds annotated with some activities. And you could use these databases to build models that allow you to predict the bioactivity for your compounds of interest. Now, the vast majority of these compounds that are included in the Campbell and the PubChem Bias database, they are simple compounds. They are what you would generally consider as drug-like compounds, something that is kinds of molecules that you see down here in terms of size in particular. And now if you're in natural products research, you're often actually exposed to compounds that look larger, that are just more complex. And our question is, can we use computational methods to predict the targets of large molecules, complex molecules, based on less complex molecules? So because for those we have lots of data available, and for those the data is very scarce. And this study is now not limited or focused on natural products, but the motivation comes from that. So in order to basically test this, we followed a molecular shape-based approach. So we basically used ROCs, the screening engine ROCs, to encode these molecules, the molecular shape of these molecules, and compare it. And by comparing the shape, aligning the molecules, and comparing the overlap, we basically measure the similarity between two molecules and associate the biological activity based on the known biological activities of structure-related molecules. So it's a very simple similarity-based method for target prediction, with the trick that you have a very robust and abstract representation of the molecular structure in the form of molecular shapes that are chemistry aware. So you don't only have the shape of the molecule, but you also have properties such as, for example, hydrogen bond donors, hydrophobic areas projected onto these surfaces or shapes. So what we tested this approach in was on 28 selected proteins. And these proteins or targets, they were selected in mind. So what we had in mind when we selected those is we wanted to cover the pharmaceutical relevant target space. So we want diverse proteins, but we want also proteins that have substantial amounts of data available on, in particular, these complex types of molecules. So for each of these 28 proteins, we selected 10 representative molecules as query molecules. And these representative molecules are all complex. This means they are large, sterically challenging small molecules. And we screened those against this large knowledge base of 300,000 molecules that are represented by up to 400 conformers. And And this database covers a total of 3,600 proteins. So for each of these queries, a total of 280 queries, because we have 28 proteins and 10 queries, we get a target list, a list of 3,642 proteins, and they are rank ordered. And what, of course, we want is the correct target for each of these queries at the first rank. And now we have a look at how far we can get with that. So this is basically a curve that shows you the success rate, the percentage of queries. So we have here the 280 queries, and this is the percentage of queries for which we have the correct target ranked among the top X or top K proteins. And these are the top 10 proteins, top 100 proteins, and so on. And remember, we have about 3,600 proteins here. And it's quite interesting to see that with the best scoring function, so this is the best scoring function in ROX because it considers the shape and the chemistry. With this one, we can basically get the correct answer, the correct target for 20% of the queries at the first rank, at the top rank. And if we allow a bit, a bit of a larger hit list, like 20 targets, then we can make it to 40 queries. So 40% of the queries have the correct target predicted among the top 20 targets. Now you may think, what does this help me if my target is one of the top 20? Well, in reality, it can be very helpful because you have some background knowledge of your queries. You know basically what your molecules do in a cell, and you may also have some additional observations. You may also not just have a single query, but maybe 5 different queries. And if you pull together these results, you may be able to basically reduce these 20 candidate targets to maybe 1 or 2 or 3, and then have some very nice working hypotheses and line avenues to proceed with your research. What we see here is that the success rates are actually very high if you use 10 molecules or so. If you use 10 molecules or so, then we have only 1, 2, 3, 4 cases where we don't have, at least in one case, the known target ranked on the top 1 position, so in the first position. So in other words, this means if you have more than one query, if you have several queries, you're likely to get actually the correct target predicted at a very high position. And the nice thing is that this method is not dependent on the size of the molecules. So what we see here is that our success rates are stable even as the size, the difference in size between our query molecule, our molecule of interest, and the nearest molecules with known bioactivity in our training or reference data set, as this increases. So this means that basically this method is really able to identify a subspace in this molecular shape, fragments, so to speak, that are decisive for the biological activity. And on the right-hand side, the panel shows that these molecules that are correctly predicted, they can be actually fairly dissimilar from anything that is known present in the reference data set. So this is the similarity here between the compound of interest, of which I want to know the target, and the nearest neighbor in the test set. And you see that even if the molecules are structurally very diverse, something like they have an 2D molecular similarity based on fingerprints only between 0.3 and 0.5, then we have already success rate in the range of 60 to 90% or so. So that's really a promising result. So the lessons that we learned from that is that, as a conclusion, 1/3 of these 280 complex molecules, we could get the correct target identified among the top 5 positions out of 3,600, and that is really an encouraging result. these 3D molecular shape-based methods, they hold the promise to identify distance similarity. And on top of that, you get these tidy overlays that you can interpret, and you nicely see the overlaps of hydrogen bonds, for example. And the interesting or good thing is also that the final predictions, they rely on a single data point, which is the nearest neighbor actually in the data set. And you can then easily go back to the literature and confirm the validity of this data point, or make a judgment of how likely your prediction is correct based on the likelihood of that the reference data point is correct. And this, we have also seen that this can even work for macrocyclic compounds, although this is a more challenging task to do. And I think in order to keep somehow with the time, I think I will conclude my talk here. I thank you very much for your attention, and I look forward to the discussion. Thank you.